Every cell needs a barrier to keep out what is supposed to stay out, to let in what is supposed to be let in, to keep in what's supposed to be kept in, and to let out what the cell doesn't want. Cell membranes serve as this boundary. Well, this separation is absolutely crucial for a cell to remain functional and alive. I hope what you will see in this video is that cell membranes, even in the most basic life, are extraordinarily complex. Cell membranes, and again, even in the most basic life, are made from compounds called phospholipids. Phospholipids consist of two long-chain fatty acids, glycerol, phosphate, and an alcohol such as choline, ethanolamine, serine, glycerol, or inositol. Carbon chains can be anywhere between 12 to 24 carbons, but usually they are 14, 16, or 18 carbons long. A different length of a carbon chain will change the properties of the phospholipid. An unsaturation, that's a double bond or a triple bond between two carbons. Unsaturations are possible between each of the carbons along the chain, and each new unsaturation would also change the properties of the phospholipid. The type of alcohol that's attached to the phosphate will also change the properties of the phospholipid. So with several different alcohols possible, several different lengths of carbon chains possible, and a large number of different unsaturations possible, a phospholipid can take on a wide range of different properties. One property they all have in common, though, is that phospholipids have one end that interacts with water and one end that doesn't. In a cell membrane, the phospholipids are aligned in what's called a bilipid structure, with all the carbon chains pointing towards each other and all the phosphate and alcohol ends pointing out. This means that if here's your phospholipid bilayer, this means that outside and inside of the cell can both interact with water or something polar or something charged, but the middle part of the lipid bilayer, where the long hydrocarbon chains are, well, that doesn't like to interact with water, making it very difficult for water to pass through a cell membrane. Polar things like water will stay inside or outside of the cell, but nonpolar things can get into the middle part of the bilayer. All cell membranes, even again with the simplest known life, have this bilayer phospholipid structure. They all also have very specific proteins embedded into the cell membrane to control what goes in and out of the cell. Some of these proteins shuttle molecules across the cell membrane. Some form selective pores or channels through the membrane. Many of these proteins are actually machines that do more than just provide an opening for stuff to come through. One example is aquaporin. An aquaporin protein allows water molecules through, but it's not just a wide open channel. The reason it can't be a wide open channel is that if water molecules come through the pore perfectly aligned, then protons, which are positively charged hydrogen atoms, so if water comes through the pore perfectly aligned, then the protons could come through at the same time with the water acting similar to a wire. If protons could come into the cell in this manner, the cell would lose its main source of energy and would die. So what the aquaporin does is rotate each water molecule in 90 degrees as it goes through the channel, creating kind of a charge barrier that keeps the protons from coming through with the water and killing the cell. Now, not all of the embedded proteins are gates or channels. Some act like receptors that allow the cell to communicate with the environment. Some catalyze chemical reactions inside and outside of the cell, and some impart structural integrity to the membrane. All of the embedded proteins must interact with the lipids. This interaction is highly fine-tuned because the lipids that are next to the embedded proteins must have hydrocarbon chains that align perfectly along the edges of the proteins to match their shape. So in that case, the protein has to fit snugly into the membrane so there aren't any leaks. So something like this, if this is the protein, and here's your phospholipid, the long chain hydrocarbons can get unsaturations in them and get kinks in them, and then they can snug up next to that protein and make it so there's not a gap so the membranes aren't leaky. Lipids and proteins have to interact with each other to have a working cell membrane. Here we have two different parts coming together into one system each part of the system looking like it was designed perfectly for the function it performs. This looks exactly like something we would engineer, but it's engineered to a degree far beyond what a human mind is capable of. But even these highly engineered lipid-protein interactions are only a small part of the complexity of a cell membrane. The organization of the lipids in a cell membrane is a highly structured system. A cell membrane is not composed of just one kind of lipid. Instead, the composition varies throughout the cell membrane, and it's not random. Remember, the alcohol and the carbon chains determine the properties of the lipid, therefore also the property of the membrane at the location of that lipid. Some quick examples. If I have shorter carbon chains or more unsaturations, that's more kinks in the carbon chain. So shorter with more kinks in them, well, that makes the membrane more fluid. But longer carbon chains and less unsaturations, that's a straighter carbon chain. That produces a more solid area in the membrane. So a cell membrane is comprised of several different domains, each structurally different in terms of the lipids, and therefore functionally different in terms of what that part of the membrane can do. The composition of the phospholipids within the membrane is designed and fine-tuned to create very specific domains within each cell membrane that correspond to the function of that specific part of the membrane. And there's even evidence that the properties of the membrane at certain locations in the cell correspond to the needs of the cell at that same location. Protein gates and non-symmetric but organized microsystems of lipids by themselves make the cell membrane a complex system. But there's one more layer of complexity. Immense numbers of polysaccharides or sugars or carbohydrates stick out of the cell membrane. These are known as glycans. The glycans are crucial for cell regulation and cell communication. All cells we know about, again, even the simplest ones, have a vast array of complex information-bearing glycans covering the surface of their membranes. There's more complexity, which we could also call information, encoded in these glycans than in our DNA. These glycans must also communicate and interact with the lipids of the membrane. So the phospholipids in a cell membrane are involved in a legion of biochemical processes. Other molecules that have opposite polar and nonpolar ends but are simpler than phospholipids, like fatty acids, they can't do this. The simplest life must have a large number of different types of phospholipids, all organized in very specific groups and in very specific locations in the membrane. The simplest life needs specific proteins in specific locations, doing specific jobs, and interacting with specific phospholipids. The simplest life needs carbohydrates, the glycans, attached to the surface of each cell, providing information about the environment and communicating with other cells, operating in conjunction with the phospholipids and the proteins. So a cell membrane of, again, even the simplest life is a coordinated, complex, engineered structure made up of many parts, each perfectly suited for their task, all communicating with each other and working together as one membrane. So, why is this important? Well, the only thing that we know of that's capable of designing and implementing an engineered, multifaceted, purposeful arrangement of parts like the cell membrane is a mind that is capable of making free will decisions, or a person. This would mean that the most fundamental fact in the universe is a person. Personhood came first. Christians believe that this person is God and that the universe was made by God for his own reasons. He engineered the cell membrane so that life could exist. Think about the foresight, the planning, and the effort that went into a life form, that went into the functioning of a human body. God had to plan from the beginning for you to be here right now. This gives us intrinsic and objective value. God was working and planning from the start to make you possible. This gives us meaning. This gives us purpose. Meaning and purpose that are grounded in God's reasons for creating us. So we have value, we have meaning, we have purpose that's independent of what we're able to accomplish or what we're able to do or not do. God's reasons for creating the universe and creating us culminated in the person of Jesus. Jesus came to earth to mend the fractured relationship between us and God. Because we constantly break God's laws, we're separated from God. So God provided a way to repair this relationship through the person of Jesus. God, the creator of the universe, wants a relationship with you. And you can have that relationship through the person of Jesus. So the exquisite fine-tuning of the cell membrane, looking as if it's designed specifically for us to exist, can point us to the person of God. God cares about you. He cares about you so much that he wants to have a relationship with you, and you can have that through the person of Jesus.